Hello and welcome to my video. In this video I will begin the discussion on mutations in cancer. This is necessary before the discussion on the protein. Scientists have found around 30,000 genes in the human genome, and out of all 30,000 genes, 291 are involved in cancer. At random? By chance? Carcinogens always hit this 1%? Before I go on, I want to give you a quick overview of genes and mutations. Inside our cells we have a nucleus, a large organelle containing DNA. Our genes are situated in our DNA. In the nucleus we find the chromosomes. If we unravel the chromosomes, we find that the DNA strands are wrapped around histones. Histones are alkaline proteins that package the DNA. And again, if we unravel the strands of DNA, we will find the genes. Some scientists are saying that mutations are alterations in the sequence of the DNA, and that if it becomes permanent, it will affect the information in a gene that might be situated in that area. In the scientific literature, we find that it is said that mutations are caused either by miscopying errors in DNA replication or due to damage by mutagens. Mutagens can be chemicals such as pesticides, preservatives in foods, or they can be high-energy electromagnetic radiation, for example UV light and X-rays, or infectious agents, viruses and bacteria. And we are told that some mutations can be repaired by the cell's repair mechanism. Some cannot be repaired. Now remember that the DNA is wrapped around histones. How could a mutagen be able to damage the DNA without unraveling the genome? Especially in cancer, the histones would need to be unraveled for the mutagen to hit the DNA anywhere, and never mind the 291 cancer genes. The only way to change the DNA through doing damage, which might result in a transformation of a normal cell, has to involve unraveling the DNA. There is no other way. Unraveling DNA has only been studied in vitro, that is, in a test tube or a petri dish, outside of a living organism, not in vivo. Even in vitro, the mutagens would have to hit the specific 291 genes. And even more than that, they would have to hit the hotspots within the relevant gene and do so by chance. This can't happen even in a test tube. It is impossible in a real-life setting. Their arguments about mutagens doing damage are all hypothetical. Fly to the moon on butterfly wings. We are told that these are random mutations, that they have happened by chance. Of all these mutations, the ones that the scientists consider lead to either a loss of function or a gain of function in the proteins made from the mutated genes. Those genes whose alterations give gain of function effects are called oncogenes, and the altered genes that give loss of function effects are called tumor suppressor genes, because without the alterations they are believed to make the proteins that suppress or prevent tumors from forming. These are the so-called driver mutations that they say lead to cancer. The claim is made in a way that is advantageous to their narrative, that cancer is abnormal cells dividing out of control, that is, uncontrolled cell growth. Have such random mutations transformed a normal cell into a malignant one? Of course not. Depending on the type of cancer, many thousands of mutations are seen in the cancer genome. And even in people with the same type of cancer, many different variations are seen from individual to individual. Thus, they require more and more mutations, and especially inactivating mutations in genes that maintain genetic integrity, in order to have an oncogenic effect. This is why they arrive at a clonal evolutionary theory. So there's Darwin to the rescue, because then they can say there is an acquisition of mutations, and having got that, there is an expansion of dominant clones. Mutations that are considered to have a selective advantage with respect to fitness are called driver mutations. These are the ones that are said to promote cancer development. Those that are seen to do nothing for fitness, but they may occur in the genomes of the same clones, are called passenger mutations. The mutated genes with these driver mutations are called cancer genes. The actions or the modified functions of the mutant proteins that are made from the mutated genes are uncontrolled cell growth, turn off any cell differentiation, avoid cell death, namely apoptosis, or will no longer mediate cell repair. All of these so-called modified functions also occur in wound healing and tissue regeneration. There isn't anything aberrant about this in wound healing or tissue regeneration. Here we have transient inhibition of p53 in wound healing. Up close for you. The main parts I have underlined in red are: in vivo inhibition of p53 accelerates early epithelialization and neovascularization of cutaneous wounds, increasing cell proliferation and reducing apoptotic cell death. So we have a situation where, on the one hand, scientists are interpreting a mutated gene as a gene that causes cancer. However, on the other hand, they are also implying that it can't be simply a chance mutation. We need to look at all the so-called cancer genes and see what parallels there are between cancer on the one hand and wound healing and tissue regeneration on the other. I am considering to make a video to do this, but it may be a while before I can finish it. A mutation is better said a gene variant. The narrative is mutagens damage DNA. However, why are the damaged genes functional? Because they are gene variants, that's why. Gene variants and protein isoforms, protein variants, is where the truth lies. I will continue the discussion on mutagens in cancer in the next video. Thank you for viewing my video, and here is a disagreement Australian bush turkey style.